I think this is very exciting news. RSV was discovered almost 60 years ago, and until the recent couple of years, we have nothing really effective that can achieve RSV prevention in the whole population. For years, we have palivizumab. That was an effective approach to prevent severe RSV disease, but only on the high-risk children. Now we move to a totally different era. We have now 2 monoclonal antibodies, nirsevimab and now clesrovimab, as well as a maternal vaccine that are all indicated to prevent RSV disease during the first season. Why is so important to prevent RSV disease in the first season? Is because it's the time that children are more vulnerable and can develop more severe disease. When you look carefully at the data accumulated over the last 20 years, you see that clearly the kids under 6 months are at the higher risk for hospitalization. That means that they can be very severely, even needing ICU care. Go beyond that. If we look beyond the more developed countries and we look at low middle income countries, RSV represents the second cause of death for infants across the world. Only malaria causes more deaths. So the excitement to have new monoclonal antibodies and a maternal vaccine to prevent this is remarkable for those of us who've been in this field for a long time. Obviously, having more than one option is very important. Why is that? Because we can target the virus in different areas. It's important to mention that nirsevimab targets what we call the site 0. If we think that the structure of the RSV fusion protein in the pre-fusion conformation is like this, very simplistic, the nirsevimab hits here, the epitope on top, while clesrovimab hits more on the bottom, right, the site 4. So this gives us more options. I think the data is we have seen published and now FDA approval is quite remarkable, and it adds an extra tool to our opportunities to prevent severe RSV disease. And you can ask me why? Well, every year in the world, 140 million babies are born, and we need to prevent RSV in as many as we can. So having this new monoclonal adds to our new tool to do that. It's a little bit different, this monoclonal, from the previously approved one, because it targets, again, a different site of the molecule, the site 4. We use one uniform dose across, so you don't need to adjust for weight, which is very interesting. And so this is really great to have this monoclonal together with the previous one, nirsevimab, and the maternal vaccine. So it's for us in the field and for those who are studying RSV for a long time is really excellent. First, the efficacy that we have seen in the clinical trials is probably better than we expected 4 or 5 years ago. So this is really remarkable and the beauty that we have seen with both nirsevimab and the maternal vaccine, when you put this in the real world evidence, also this holds on. So the expectation is that when we do the real world evidence with clesrovimab will be very similar to the numbers that we seen in the clinical trials. And this is very critical because if we look at the time that babies get hospitalized, the peak is between 1 and 2 months. So these are very early, right? So the people sometimes ask me why we don't have an RSV vaccine. Well, number one, RSV affects babies very early on, right? So even if we start vaccinating at 2 months, we are going to miss a number of cases. But second, we learned that RSV has certain genes that makes the immune response poorly effective against RSV in a natural setting, right? So we learned that maybe the idea of vaccinating at typically 2, 4, and 6 months that we use for pertussis or for Haemophilus influenza might not be the best strategy for RSV. And for that reason, all these studies have been done what we call passive immunity. That means that we give an antibody, pre-developed antibody, to the infant. So the infant is protected from that time. So this could be done either through a monoclonal antibody, right? We give them monoclonal to the infant, or by immunizing the mom the antibodies cross the placenta and also protect. So we are giving the antibody already developed, instead of as in a regular vaccine that we give like a signal for the baby to make their own antibodies response. That will not work for RSV. So these three strategies are really positive. Another important detail, some data suggests that if the babies get infected in the first 6 months of life, the likelihood of having more recurrent wheezing, recurrent respiratory infection is much higher. So we have expectations that with these new interventions, with the monoclonals and the vaccine, maybe we can not only prevent the acute events, but maybe have a potential long-term benefit in preventing other diseases like wheezing or even asthma, right? We don't know that yet. Those are the studies that we need to do. Now, this has been the association between RSV and recurrent episodes of wheezing has been studied for a long time. But some new evidence is beginning to show that also RSV interacts with bacteria, specifically with pneumococcus. There is direct evidence that a protein of RSV, RSVG, binds to PVP1A of pneumococcus. And that facilitates the pneumococcus infection. The pneumococcus is more virulent, the pneumococcus attached to the epithelium and causes possibly pneumonia. So we hope that one advantage could be that maybe we can also prevent some of the pneumonia.